So good evening students. Today we will see a short video on excretory physiology. The important topics I will try to cover. The first is the functional anatomy. You should know the difference between the cortical nephrons and juxtamedullary nephrons. The cortical nephrons, the major role is in the form in glomerular filtration, and this juxtamedullary nephron, the main role in the concentration of the urine. The cortical nephron constitute about 75 to 80% of the nephrons, and the juxtamedullary nephron constitute about 20 to 25% of the nephrons. And the loop of it will be short in cortical nephrons, and in the juxtamedullary nephrons it will be longer. And coming to the urine formation, the urine formation is having three basic steps. One is the glomerular filtration, other one is the tubular reabsorption, and other one is the tubular secretion. The glomerular filtration is the amount of filtrate or ultrafiltrate of plasma that is formed by both the kidneys per unit time, and the normal value is 125 ml per minute or 180 liters per day. And for the filtration to occur, the very important membrane is called the filtration membrane, which is made up of the capillary endothelium, the basement membrane, and the epithelium of the Bowman's capsule. And what are the determinants of the GFR? One is the hydrostatic pressure, that is the capillary hydrostatic pressure, capillary oncotic pressure, tubular hydrostatic pressure, and tubular oncotic pressure. So now you group the filtration depends on the net filtration pressure, which in turn depends upon the factors favoring the filtration minus factors opposing the filtration. The factors favoring the filtration are the capillary hydrostatic pressure and tubular oncotic pressure, and factors opposing the filtration are the capillary oncotic pressure and tubular hydrostatic pressure. And when you put the values, we'll get the net filtration pressure as plus 10 millimeter of mercury. So the GFR is directly proportional to the net filtration pressure, and it is proportional to Kf also. So what is Kf? Kf is the filtration coefficient. This Kf depends upon the capillary permeability and the filtration surface area. And the permeability of the capillary in turn depends upon the size of the particle and the charge of the particle. If the size of the particle is less than 7 or 8 nanometer, it will be filtered easily. And if the charge is positively charged, it will be filtered through the negatively charged basement membrane. So here one question is why albumin, the size is 6 nanometer, is not filtered? Because albumin, though the size is less, here the charge of the albumin is negative charge, so it is repelled, so it is not able to filter through the membrane. Okay. So now what are the factors regulating the GFR? One is the tubular glomerular feedback. That is when there is more filtration, there will be more sodium delivery to the distal tubules, and there is a sensor called macular densa which sends the signals, and it will cause us the alteration in the afferent arteriole and efferent arteriole. So let us start with the flow chart. Like when there is a decrease in the GFR, there is a decrease in sodium delivery to the distal tubule, and this was sensed by macular densa, and it sends signals to the JG cells, dextroglomerular cells, to secrete renin, and this renin causes efferent arteriolar through angiotensin 2. So renin causes angiotensin 2 to be produced, and this angiotensin 2 will cause efferent arteriolar constriction, and this increases the capillary hydrostatic pressure and increases the GFR, and again the sodium delivery will be normal. So this is one way renin angiotensin mechanism that is auto-regulating the GFR. The other one is the myogenic theory, which auto-regulates the renal blood flow. So more the renal blood flow, more will be stretching of the smooth muscles of the blood vessels, and this will cause contraction of the smooth muscles, and this will reduce the renal blood flow, and thereby regulates the GFR, because GFR depends upon the renal blood flow. And third is the hormones. So the hormones, especially the norepinephrine, epinephrine, these hormones causes afferent arteriolar constriction, and thereby it reduces the blood flow, and it reduces the GFR. And another one is the hormones like nitric oxide and prostaglandin. They causes afferent arteriolar dilation, they increase the blood flow to the capillaries, and they increases the GFR. So these are some of the factors that regulate the GFR. And next is, after the filtration, the next process is the tubular reabsorption, and various solutes are reabsorbed in various segments. So we will start with the renal handling of sodium, how sodium is reabsorbed. So once it is filtered, about 60 to 65% of sodium is reabsorbed in the proximal convoluted tubule. So it is a segment where maximum absorption of solutes takes place. And after that, the same and again in the thick ascending limb of loop of Henle, sodium is transported into the medullary interstitium, is actively absorbed by sodium potassium 2 chloride sympone. Then in the distal tubule also sodium is reabsorbed. So these are the various areas sodium is reabsorbed. And coming to the H plus ion, this we call it as renal handling of H plus ion, which is very important for acid base balance. So H plus ion is normally it is exchange for sodium reabsorption in the proximal tubule. In proximal tubule, what happens? Sodium is reabsorbed and H plus ion is secreted. So whenever the EC of H plus ion concentration increases, this mechanism is very active, and it causes H plus ion secretion in the tubular fluid. And the H plus ion is also actively pumped out in the collecting duct by H plus ATPase pump, which is present in the intercalated cells. So the intercalated cells has H plus ATPase pump that pumps the H plus ion. In both these conditions, the H plus ion which is secreted combines with bicarbonate and converted into bicarbonic acid, and which dissociates into H2O and CO2, and CO2 again enters into the tubular epithelium, and there it is again transported into the tubular into the blood. Okay, so for every H plus ion, one bicarbonate will be reabsorbed. Because in the tubular epithelial cells, what happens, the carbon dioxide which is entering combines with water and forms carbonic acid, will dissociate into H plus and bicarbonate, and that bicarbonate will be reabsorbed into the blood. So whatever filtered bicarbonate, it is reabsorbed in another form of bicarbonate from the tubular epithelium into the blood. So for every H plus ion secreted, one bicarbonate is reabsorbed. Here it is not the same H plus ion, which same bicarbonate which is filtered is not reabsorbed. Okay, it is a different bicarbonate. But anyhow, for one H plus ion which is secreted, one bicarbonate is reabsorbed. Now, similarly, in the correct also the same process. H plus ion, which is pumped out by H plus ATPase, combines with the carbonic bicarbonate, which is filtered, and forms carbonic acid in the tubular fluid, which is associated to H2 and CO2, and this CO2 enters into the epithelial cell, where it combines with water. Again, the same process, carbonic acid is formed in the epithelial cell, which is associated into H plus and bicarbonate, and that bicarbonate is reabsorbed. So this is the visualization of the process taking place. And this H plus ion here in two areas it is secreted. Okay, one is in the proximal tubule, and another one is the collecting duct. Also, in the collecting duct, H plus ion is also combining H plus ion in the form of ammonium, which is a product from the glutamine. See, glutamine on metabolization gives 2 bicarbonate and 2 ammonium ion in the collecting duct epithelial cell, and this ammonium is diffusing into the tubular lumen, and it combines with chloride ion. So in the ammonium we have H plus ion, which is eliminated from the body, and this buffering by the phosphate by the ammonium is called ammonium buffer. This is called ammonium buffer. It plays a very important role in chronic acidosis. And another buffer is phosphate buffer. So the phosphate buffer is the phosphate which is filtered, which is called a sodium dihydrogen phosphate, and this phosphate combines with H plus ion in the collecting duct, where H plus ion is pumped out, or in the proximal tubule where H plus ion is coming, it combines with the phosphate and forms disodium hydrogen phosphate, and it is eliminated. So H plus ion is eliminated in two buffers. One is the phosphate buffer, other one is ammonium buffer. So these buffers plays a very important role in the elimination of non-volatile acids. See, volatile acids can be it is a bicarbonate, the bicarbonic acid, which can be eliminated by the lungs also. But non-volatile acids of 80 milliequivalents is eliminated mainly by these buffers. So these buffers, for the elimination of these buffers, more H plus ion is secreted. Okay. And this occurs above the limiting pH also. So this is very important, and this acid which is eliminated is called the titratable acid. And so these are the H plus ion handling by the various segments of the tubular lumen. Then we have the water. So water reabsorption, water is more permeable in the descending limb of loop of Henle, and water is also absorbed in the cortical collecting duct in the presence of ADH by acting on the aquaporin channels, and it is also in the medullary collecting duct also in the presence of ADH. Water is actively reabsorbed through the aquaporin channels. Okay, it is water is reabsorbed. Next is the urea. So the urea is actively transported by the urea transporter present in the collecting duct, and this urea helps in increasing the interstitial medullary osmolarity along with the sodium and chloride ions. And this urea, some amount is entering to the thin ascending limb. This is called the urea recycling. So urea which is absorbed into the interstitium from the collecting duct, again entering to the tubular fluid through the thin ascending limb of loop of Henle. Now these are some of the renal handling of the various solutes and water. And coming to the countercurrent mechanism is a mechanism that helps with the concentration of the urine, and here we have two structures involved. One is the countercurrent multiplier, other one is the countercurrent exchanger. The countercurrent multiplier is the loop of Henle. So what happens in loop of Henle, the solutes which are filtered from the in the glomerular filtrate, these solutes they enter into the thick ascending loop of Henle, and in the thick ascending loop of Henle they are actively reabsorbed. The sodium chloride and potassium is actively reabsorbed in the interstitium, and they increase the medullary interstitial osmolarity. So this medullary interstitial osmolarity is a very important factor for concentration of the urine. And whatever solutes is entered into the medullary interstitial osmolarity, it makes water to move from the thin descending limb by osmosis. Now what happens in the thin descending loop of Henle, water is reabsorbed by the process of osmosis, and the solute concentration will be increased, and these solids again will come to the thick ascending loop of Henle, again it is reabsorbed, and this causes movement of water from the thin descending limb, and this process happens in a cyclical way, and the urine which is the tubular fluid which is leaving the ascending loop of Henle, it is more hypotonic, okay? And when it passes through the descending limb, again the sodium and chloride is reabsorbed, and it will be still more hypotonic, and this very hypotonic urine when it comes to the collecting duct in the presence of ADH, enormous amount of water is reabsorbed. So how it is happening is because of the medullary interstitial osmolarity. So two factors helps in the water movement from the collecting duct. One is the medullary interstitial osmolarity, other one is the ADH, and these two factors makes water to move from the interstitium, from the tubular fluid into the interstitium, increasing the urine osmolarity even up to 1200 milliosmoles. This is the maximum concentrating ability of the urine. Also, when this movement is happening, the urea concentration will be increased. Since water is moving, urea will increase in the collecting duct, and these urea also move into the interstitium by the action of ADH on the urea transporters. And when urea moves, again water will be moved. This is called solvent. This is the urea is making the water to move by osmosis. So ultimately there will be a concentrated urine be formed, and it is eliminated through the collecting duct. This is the countercurrent mechanism. So whatever solute which is coming to the interstitium again goes back to the blood vessel, that is the vasa recta, and once it enters the vasa recta, the descending limb of vasa recta, and from the descending limb water will be moving into the interstitium, and again water will be moving into the ascending limb, and again solute will be moving out of the ascending limb. This is taking place in vasa recta. So vasa recta is a specialized peritubular capillary. What to remember is this vasa recta helps to maintain the interstitial osmolarity created by the Henle loop, so that they don't get washed out. Only 5% of renal blood flow is passing to the vasa recta, and this little blood flow helps in preventing the loss of medullary interstitial osmolarity and maintaining the osmolarity. Okay, so some added points is juxtaglomerular apparatus. The juxtaglomerular apparatus is made up of juxtaglomerular cells, extraglomerular mesangial cells, and macula densa. So what is the role of juxtaglomerular apparatus? It helps in the synthesis of, it helps in the renin-angiotensin mechanism. So the JG cell secretes renin, and this renin is secreted on stimulation from the macula densa. Macula densa is a sensor; it sends the sodium load. When sodium load is less, it sends the signals to JG cells to secrete renin, and this renin converts angiotensin 1 to angiotensin 1, and angiotensin 1 is converted to angiotensin 2, and this angiotensin 2 causes efferent auricular constriction, and this causes increase in the GFR, and it causes increase in the filtration of sodium, and this is a process taking place whenever the sodium filtration is getting low, when blood pressure is getting low. So these are the renin-angiotensin mechanism, and this was effectively done by the juxtaglomerular apparatus. And so these are some of the important topics I try to cover in the excretory physiology. Okay, thank you.